Well, welcome. Thank you for joining us at this special lecture and celebration of the 2021 Rolf Medal. I'm Paul Goldbart, Stony Brook University's Provost and Executive Vice President for Academic Affairs, and I am really very pleased indeed to host this event as part of the Provost's lecture series. I'd like to acknowledge the support of the College of Arts and Sciences Office of the Dean, thank you Nicole, as well as the Departments of Anthropology and also Ecology and Evolution for their collaboration on today's terrific event. A very warm thank you to Jim and to Janice Rolf for their dedication to Stony Brook University and for making today's event possible. Jim, as I think everybody here knows, is a distinguished professor emeritus in the Department of Ecology and Evolution and a research professor in the Department of Anthropology. And the Rolf Medal, and I think this is just a wonderful, delightful touch, I really do, it was created in 2006 by Jim's family in honor of his 70th birthday. Dr. Rolf received his PhD from the University of Kansas in 1962. Fellow of the American Academy of Arts and Sciences and of the American Association for the Advancement of Science. He's organized and presented at numerous international workshops on the topic of geometric morphometrics. And to date, five experts have been honored with the Rolf Medal. 2 of them are alumni of the PhD program in the Department of Ecology and Evolution. So at this time, I'd like to turn things over to the 2019 recipient of the Rolf Medal, Dr. Dean C. Adams, class of 99 here at Stony Brook, and current chair of the Rolf Medal Committee. Thank you very much, everybody, for joining us today, and enjoy the program. Thank you. Thank you.
Thank you very much. Good afternoon, everybody. It's very nice to be back here. Again, my name is Dean Adams, and I'm an alumnus from 1999 in ecology and evolutionary biology. I, this year, had the honor of serving as the chair of the Rolf Medal Selection Committee, and the committee was comprised of myself, Eric Delson, Paul O'Higgins, Benedict Hallgrimsson, and Miriam Zeldich. And we had an outstanding series of nominees and candidates that we had to select from. And on behalf of the committee, as I need to say, it of course is my pleasure to announce that Dr. Joan Richtsmeier is the 2021 awardee of the Rolf Medal. Now, throughout her distinguished career, Dr. Richtsmeier has really pushed the boundaries of morphometrics, and in particular its application in anthropology, evolutionary biology, and biomedical sciences. Her work on craniofacial morphology and development espouses, to my mind, a whole new conceptual approach that merges morphometrics, molecular, cellular, and developmental techniques to result in a more synthetic understanding of disease etiology. She's also furthered evolutionary biology and anthropological applications by employing a diverse set of analytical and morphometric tools in her work, which has helped move anthropology in a far more quantitative direction. And within the biomedical sciences, she's been a tireless advocate of increased quantitative rigor, and in so doing has brought morphometric methods to a far broader realm of scientists and subdisciplines than it otherwise would have been exposed to. And for these reasons, and for many other reasons that the committee discussed, we were pleased to award Dr. Richtsmeier the 2021 Rolf Medal. And so at this point, I would ask Joan to please stand and receive the award. Congratulations.
First, here's the check for the past prize. Do this somehow. COVID compatible. And as I pointed out to her earlier, it's pretty heavy. She doesn't have to lecture with that on. It's too much of a burden.
Never taken it off. Thank you so much.
Hi, I'm Fred Bookstein, one of the earlier medalists. Good afternoon. The work for which Joan Richtsmeier gets this year's Rolf Medal links anthropology and medicine by bridging craniofacial dysmorphology to anthropometrics. Hers is a very good example of how geometric morphometrics, the Rolf's Medal theme, can collaborate in multidisciplinary investigations. So I'd like to take a few moments to talk about the conditions favorable to those collaborations. The core data type of today's GMM is the notion of the landmark point, codified a long time ago in Rudolf Martin's great Lehrbuch der Anthropologie of 1914. Already back then, students of human evolution and variation were tabulating interlandmark distances more than one at a time, and the extension to their joint analysis followed very soon in the Indian school of statistical anthropologists like P.C. Mahalanobis and his still living disciple, C.R. Rao. Richard Raymond, a Swede, was already promoting more general evolutionary contexts for the multivariate study of distances by 1971, half a century ago. And less than 20 years later, at the 1991 NATO meeting run by Rolf, Les Marcus, and me, the toolkit received its imprimatur for the emphasis on landmark geometry and its classic multivariate statistical analysis. Biometrics was simpler back then, without interactive 3D medical image analysis, surveillance video processing, DNA sequencing, knockout technology. What kinds of tools from this 20th century-based version of anthropometrics can fruitfully collaborate at today's multidisciplinary cutting edge? I'd like to suggest 4 principles that together account for the power of GMM in Richtsmeier's hands. 1, the real existence of predefined groups. Apert syndrome was announced by Eugene Apert in 1906, Cruzan by Octave Cruzan in 1912. The Indian races whose discriminant function C.L. Rao was computing from principal components of distances in 1948 had been officially codified during the British Raj, just ending. Carl Pearson identified the skull of Oliver Cromwell by its landmark morphometrics in 1935. Anthropologists have been describing groups or specimens by landmark locations and their distances for a very long time, and GMM embraces this older theme in full. 2, circumventing causal discussions or relying on other kinds of data for them. The existence of those groups is logically prior to anything GMM can say about how cases came to be members of them. Back when I was working on the craniosynostoses in the 1980s, we didn't have the genomics, and we didn't have the animal models, but we did have the cephalograms and the surgical experience. So the emphasis was on surgical planning, wound healing, and relapse versus post-surgical growth. We weren't asking why questions. Why does this particular family tend to hypertelorism? But we were asking how we optimally intervene to normalize forms this aberrant. The landmark templates that work for surgical planning are not necessarily helpful for those causal questions, which demand biological insights deeper than what GMM can provide. 3, restriction of anatomical information to the locations of boundaries. By this I mean the simplification of actual image contents of delineated organs, such as the morphometry of histological sections or the gray levels of CT or MR imagery, down to the curves or surfaces of sharpest image gradient, where organs can be dissected away from their neighbors or from air by automated algorithms, such as trained graduate students. And 4, exclusion of dynamical considerations. This 4th category may prove the most important for the future of GMM. Principal component analysis is grossly inadequate for studies of serial changes of landmark configurations, whether growth or evolution, and I mean this literally. The mathematics of PCA is incapable of describing the general case of sequential change of form. The foundation of this critique was already laid by the Viennese theoretical biologist Karl Pribram in the 1920s. He objected in principle to Darcy Thompson's method of Cartesian transformations. For the representation of form changes by deformation grids to be biologically meaningful, he said, you need actual data about how different grids change differently. Explanations of the curving and bulging of form in Thompson's phrase, "the origins of form in force," are unpersuasive when phrased only in terms of discrete landmark locations rather than underlying continua. But when my four constraints obtain, as they do in Professor Richtsmeier's work, excellence of the GMM can merge with excellence across all of these other coordinated aspects of organismal biology that combine in explanations of the causes of those a priori groupings and the salience of their consequences. The work for which we honor Professor Richtsmeier today aligns neatly with all four of these requirements. The synostoses are genetic in origin, and their medical surgical management doesn't much depend on the details of the contents of the volumes whose conformations are so obviously abnormal, but instead straightforwardly accommodates monogenic explanations. The genomics and the histology accord with the predefined classifications. The genes forecast what the fetus will eventually do. The histology hints quantitatively at the tissue dynamics by which this causation is effected, while the landmark analyses, likewise consistent with the classifications, are non-committal as regards either genomics or histology. Biomedical meaning comes from the medical or social consequences of the developmental causes driving the intermediating morphometric study designs. The role of GMM in contexts like these is one of confirmation, not discovery. So Professor Richtsmeier's mix of toolkits, including the role of GMM per se, brilliantly suits applications like hers that require no mathematics more advanced than what's built into the measuring instruments themselves, the anatomical scanners or spectrographs or stereometric image processors or DNA sequencers or microarrays. This application style of GMM is likely to remain central for decades to come. Extensions to this classic GMM toolkit, mathematical maneuvers much more advanced than linear multivariate statistics of coordinates or distances, transcend one or another of those restrictions for studies of selection gradients explicitly observed, or actual deformations of tissues or organs, or their function, or data like histology or genetics, or life course of the same landmark specimens. One neighboring field, medical image analysis, extends the morphometrics of the head by applying partial differential equations in studies of whole pictures in order to extract gradients and flows of their gray levels or colors. For instance, the realities of Alzheimer's syndrome are described by partial differential equations that analyze brain image changes over age by genome. Here there are neither landmarks nor semi-landmarks inside the skull. Everything in an in vivo image is grayscale. Or from another neighboring field, geostatistics, GMM can borrow a concept of integration applicable to landmark data, where thin plate splines work as prediction functions. Like shape, integration is another simple-sounding biological notion that requires much deeper math than classic multivariate statistics if it's to be implemented meaningfully in arithmetic. What Professor Richtsmeier will present today serves as a splendid example of how to execute the GMM component of a multidisciplinary study when landmark data are enough to capture the relevant anatomy across pre-existing categories, here the craniosynostoses. The methods that work so well for her deserve to be emulated wherever, as she notes in the poster for today's talk, morphometrics furthers the understanding of a group of disorders, as long as any contrasts confirmed morphometrically via discrete landmark data match the contrasts observed genetically, histologically, aesthetically, or clinically. Indeed, studies along these lines, aimed at enriching the narratives of medically relevant human variants, account for most of the willingness of countries like ours to support academic biomedical research more generally. I look forward very much to today's autobiographical journey through evolutionary morphology and then craniofacial biology to arrive at the synthesis for which this medal was awarded. Ladies and gentlemen, please welcome this year's Rolf Medal winner, Professor Joan Richtsmeier.
So thank you very much for those lovely introductions. I am incredibly honored to be here today. They told me I could take this off while I talked. Dr. Rolf said this is really heavy and I could take it off. I'm not taking this off. Thank you very much. I really like jewelry and this is a nice big piece. So thank you very much for being here today. So I'm a morphologist, and that means that I'm interested in the shapes of things. But I'm not just interested in their shapes. I am interested in how those shapes come to be over evolutionary time and over ontogenetic time through processes like growth and development. Now, morphology is sometimes thought of as a descriptive science, descriptive art, but I really feel that you have to have a precise quantitative description of shape or form before you can tell if there's been any change. And it's that change that I'm actually interested in. Change that's often driven by evolution or driven by growth and development. So it was these questions of evolution and growth and development that were swirling around in my head when I, as a graduate student, third-year graduate student, I took a job as a part-time data input specialist, which means typist, at the Center for Craniofacial Anomalies at the University of Illinois. And this was a very special place that was founded by Dr. Sam Pruzanski. It was a place where children with craniofacial deformities could come and in one visit see every specialist they needed to see, be it a neonatologist, a speech pathologist, a pediatrician, craniofacial surgeon, whomever they needed to see, that specialist would be there. And this was important for two reasons. Number one, that meant that that family that was already undergoing incredible financial and emotional hardship only had to go to one place to see these specialists. But two, and maybe most important, is that those specialists then got together and talked and collaborated about the care that they were going to plan for that child so that they could coordinate the different therapies and the different surgeries that these kids had to undergo. So because I was a data input specialist, I didn't have an office. I had a desk in the hallway. And on clinic days, children like this were either carried by my desk or walked by my desk with their families. And I got incredibly interested in the idea of how did a face like that grow? I knew about normal growth and development, kind of, but I couldn't understand how these faces would grow, nor how the growth of their face might contribute to this dysmorphogenesis that was occurring every day of their life. So this particular group of diseases are known as Craniosynostosis Syndromes. And craniosynostosis, quite simply, is a disease that can have a lot of things can be wrong with these kids, but they all shared one diagnostic thing, and that was premature closure of the craniofacial sutures. So what you're looking at here are doublets of computed tomography images of infants. The big one in the middle is a typically developing individual. The forehead is at the top, the back of the skull is at the bottom. This is a bird's eye view. This is what we call a worm's eye view. So you're looking from the bottom. This is foramen magnum, back of the skull. And here's the maxillary arcade. Typically in developing individuals, each of these bones of the skull come to be, ossify as bony, separate bony plates. And they're separated by non-mineralized fibrous tissue known as sutures. And those sutures are important for several reasons. One is it allows give and take for the bones as they pass through the birth canal. But secondly, and more importantly, these are growth centers. So new bone is laid down along the edges of the opposing bones on each of these sutures, so that as the brain is growing exponentially, those bones more or less float on the surface of the brain, with new bone being put down at the sutures. Now, a child with craniosynostosis has premature closure of those sutures. So, for example, if the sagittal suture, and that's a suture that goes from the top of your head to the back of your head, if that closes prematurely, you don't get growth perpendicular to the suture, and instead you get compensatory growth at all of the open sutures, such that you can see the closed suture. These are doublets of infants with, this happens to be sagittal synostosis, closure of that suture. You get no growth perpendicular to that suture, compensatory growth anterior posteriorly, so you end up with a very narrow skull that's elongated from front to back. If, on the other hand, the coronal suture, which stretches from one ear over the top of your head to the other ear, is closed prematurely, growth can occur perpendicular to this suture. Compensatory growth occurs at the other sutures, and you end up with no suture here and a skull that's foreshortened from front to back and wide mediolaterally. And this can occur bilaterally in that case, or it can occur on one side or the other, producing what we call plagiocephaly, or twisted skull. So I saw these kids coming by the desk every clinic day, and I was really asking myself, how do their faces grow? Well, fortuitously, one of the aspects of patient care at CCFA was that they took an X-ray of each child once a year. And I told Dr. Przansky that I was interested in this question of how the faces of these kids grow. And he said, oh, well, look. And he took me into a room that was about 10 by 15 feet, and it was filled with hard copy X-rays. In essence, longitudinal growth data for all of these diseases. So I was off and running with a really nice data set. And in those days, when you studied growth, this was long before CTs, we had these lateral X-rays. So what you did, you used this method called Brentographic Cephalometry, which simply means taking measurements from head X-rays, and you learned where the bones were. You do an outline of the bone and maybe an outline of different parts of the skin that overlaid that. And then you plotted landmarks, and then you compared two ages. So for example, here I've got a child's X-ray at age 1, at age 2, and I draw an outline of the bones that are there. I plot landmarks, and the conventional way to study growth was to register on this point sella, which is actually sitting in the center of the pituitary fossa, superimpose on a line that stretched from sella to nasion, and you looked at how those different points moved in space. And that was your data, your interpretation of how growth had occurred. Well, I knew these kids' faces were really dysmorphic, and I felt that constraining growth from this point sella to nasion, which sits between your frontal bones and your nasal bones, just couldn't really be happening, that this probably wasn't the way growth occurred. Consequently, I chose instead to register on the same point sella and to superimpose on a line that stretched from sella to basion, and that's a landmark on the front of the foramen magnum, and then interpret the movement of points as growth. Well, this seemed okay until I compared the results of these two superimpositions, and I saw that they were totally different. It was the same data, but I was getting a totally different answer. And there was nothing within these data sets that could tell me which one of these two was correct. And so I thought, I need a different method. Luckily, my advisor, Jim Cheverud, was working with a group of engineers at Northwestern University, and they had this idea to take a method that was standard to engineering, finite element analysis. Finite element analysis is usually used, you have a form, you probably know the material properties, you apply a particular force, particular known stress and strain, and it can predict how that form is going to deform under that strain. These guys, Jack Lewis and Bill Liu, turned this upside down, and they decided in biology what you usually have is an initial form and a target form, and they turned the method around so that you could apply finite element scaling analysis to a comparative analysis of those two forms, and the method would tell you what it took, what was the stress or strain, the force that was required to go from the initial to the target form. So this all came out in Lagrangian strain tensors. Jim Trevor will go to heaven for spending months with me explaining engineering to me, teaching me matrix algebra so that I could understand what the heck this method was, looking at the theory behind all of this. And so the results came out as Lagrangian strain tensors that were specific to landmarks that were connected by finite elements. This is a graphical representation of that output. If there had been no deformation at a particular landmark, it was simply a circle. This was 2D data, remember, X-rays. And what the Lagrangian strain tensor gave you was the maximum and minimum magnitude of strain and the directions. The vectors gave you the directions of that strain. So this is an interpretation of growth of a child with Apert syndrome from age 1 to 4. I also had normative data, so I could also estimate growth in a typically developing child from year 1 to 4. What the method didn't have was a way to compare those growth patterns. But we had 2 graphical outputs, and I could describe what those differences were. We could do a little bit of mathematics of comparison, but nothing impressive. So, but it was enough for me to finish my dissertation, so I handed that in. I wrote up papers for publication and was lucky enough to get a job at the Johns Hopkins University School of Medicine. This was my first academic job, and I was thrilled to be there, and I was very surprised that quite soon after I arrived there, people from the Department of Craniofacial Surgery wanted to talk to me because they had this great new part of patient care, which was creating computed tomography images of kids with craniofacial anomalies. And this was the first time I got to see what a suture looked, what it was closed prematurely, got to see what the cranial base looked like, this part that I was supposed to assume, grew linearly. And I was really excited about it. But the reason why these surgeons wanted to see me is because they wanted advice on when to operate on these kids. They wanted to know specifically how these kids were growing, what the timing of it was, and when was it going to be a good time to plan different operations. And with these types of questions, and I knew I couldn't afford to be wrong. Meanwhile, reviews of the papers that I had submitted for my PhD were coming in, and they were devastating. I had clinically oriented reviews that said, how could this person, I think they said girl, but how could this person who's an anthropologist look at an X-ray and understand what bones she was looking at? And so I'd studied osteology with the great Jane Bijkstra. I'd been taught to read X-rays by Sam Przansky and how to interpret them. But so I really thought I could handle those reviews. But there were also reviews on the use of finite element scaling analysis, and the reviews were filled with equations, and they were written in a way that I didn't really understand. I didn't know what the reviewer wanted me to do. And so being relatively new to Hopkins, with few close colleagues, and no co-authors, I sought help outside my discipline. So I called up the chairman of the Department of Statistics at the School of Health and Hygiene, told him the situation, and being the lowest person on the totem pole in the Department of Cell Biology and Anatomy, I was introduced to the lowest person on the totem pole in the Department of Statistics, and that was Subhash Lele. I took my reviews over to him. I told him about finite element scaling analysis. I told him about Brentographic Cephalometry. I said, I don't understand these reviews, and he listened to me, and then he sat back in his chair, and he said, but Joan, what's your scientific question? He wasn't interested in the details of the reviews. He wasn't even interested in the methods that I had been using. He was interested in the scientific problem that I had defined and how I was asking that question. And this made me really have to think about what is it that I'm trying to accomplish here? What is it I want to know? and I decided that I wanted to know how the heads and faces of these kids grow. I wanted to know whether or not that growth pattern was different from growth pattern of typically developing kids. And I wanted to know whether or not this growth pattern contributed to the dysmorphogenesis of these kids. So what I really needed was a method that could statistically compare comparisons. And so I taught Subhash biology, and he taught me statistics, and together we developed a method that we named Euclidean distance matrix analysis. And this method is based on the principle of invariance. It's based on a really simple idea of getting rid of the coordinate system, because the coordinate system causes problems. So what we did was we rewrote the description of form that was collected as landmark coordinates as a group of unique linear distances among those landmarks. This linear distance data, we called it the form matrix, and it's a description of form based on landmark data that's invariant under translation, rotation, and up to a point reflection. We provided a test for the hypothesis of similarity in forms. We provided a statement of variance of estimation through the estimation of confidence intervals, and a method for comparing growth patterns. So this is really on this slide all there is to EDMA. It's really an alternate approach to dealing with 3D landmark data. So Subhash worked on all the theory. I was responsible for the biology. Tim Cole, who's an illustrious graduate of this very institution, he was a postdoc in my lab, he wrote some really creative auxiliary EDMA routines and graphical approaches. He wrote the first programs that were in the Windows environment, and we put this all together in a book. And more recently, with some new scholars, we have tried to improve the variance-covariance estimate, and Peter Salimos has now put this all together in a program, EDMA and R, which is available on GitHub. So the idea behind the method is really quite simple, but from a biological perspective, it's really powerful. And the strength, in my opinion, of this method lies in its ability to localize differences between forms through this bootstrap approach. So for example, if I want to compare growth between two individuals, or form between individuals here at age 1 and age 2, you can have linear distances between two ages that get larger with growth. You can get linear distances that remain the same in length, and you can get linear distances that actually are reduced in length during growth. And that's when bones rotate on one another during the growth process. This happens specifically in craniofacial growth. But there was extreme pushback from the morphometrics community about this approach, and the reviews of my work submitted for publication became very personal and very mean. Every study for me was a fight to publish. When I fledged trainees and students, they got the same kind of reviews of their own work that actually mentioned me by name. So I was fighting my own wars, trying to get tenure, trying to get everything published. And because I felt, really responsible for my fledged trainees, I was helping them to try to get published too. And this was exhausting, and it was demoralizing. And I realized that at that point in time in morphometrics, there really wasn't room for more than one approach. And this isn't really healthy for any scientific discipline, and it wasn't healthy for me. So I moved on. And it was in this environment of developing a method for studying growth, trying to understand the new molecular knowledge about craniosynostosis, and working out how to deal with 3D data from these computed tomography images that I wrote my first NIH grant. And in those days, they still do have program project grants, but program project grant is something where a number of PIs get together and they write a grant about a generalized topic. So we were writing a grant about craniosynostosis. There were 5 projects, 3 cores. I think there was a molecular core, a clinical core, and a statistics core. 5 projects. In those days, if your grant got through the first review, they organized something that's called a reverse site visit, where the NIH came to your university. So the 5 of us, 5 PIs, gave presentations in an auditorium like this to 35 reviewers, one at a time. And then we were escorted into a room to have the review panel decide our fate. So we're sitting in that room, and there's a knock on the door. Dr. Richtsmeier, the review committee would like to talk with you. Well, I knew this probably couldn't be good. So I went back into the auditorium, and one of the reviewers stood up and said, Dr. Richtsmeier, you're in a program project grant with 5 projects. 4 of them are molecularly based. Yours focuses on morphology. What can you possibly learn about craniosynostosis by studying morphology? Well, first I was floored. I didn't know what to say because it is morphology. It's the phenotype that has to deal with the world on which natural selection operates, which is the source of teasing by unkind classmates. That's the material operated on by craniofacial surgeons. What do you mean? What can morphology teach you? But I didn't say that. I knew I was going to have to take a deep breath and explain to these reviewers who felt that genetics was the answer to everything what we could learn by studying morphology. And the thing I learned from this experience was to always be ready with an elevator talk that explains your work succinctly and also explains why it's important. I got through this elevator talk, our grant got funded, and this was a light bulb moment for me because I realized that what I was studying was morphology after the fact. By studying postnatal growth, I was studying a process where the damage was already done. I was able to describe change quantitatively, but I was unable to define any mechanism. So I decided to write an NIH fellowship to study genetics. I didn't like genetics. I had to take genetics as a graduate student and actually kind of found it boring. It wasn't genetics that I was interested in. What I wanted to understand was what mechanism meant. I couldn't understand how the identification of a gene could tell you about mechanism. So I got one of these Ruth L. Kirstein fellowships to study genetics with the great Roger Reeves at Hopkins. Roger's a geneticist who's interested in gene dosage imbalance. And one of the diseases that's a gene dosage balance disease is Down syndrome. And most of you know that Down syndrome is caused by having 3 copies of all the genes on chromosome 21 present. Roger had a mouse model, the TS65DN mouse model, which carried all of the orthologs that were on chromosome 21, happened to be on mouse chromosome 16. They were all present in this mouse model. And he had already demonstrated that these mice showed behavioral and cognitive deficits that were consistent with what we knew about Down syndrome. But the public was not accepting this as a model for Down syndrome. And Roger thought that if we could, there were 2 characteristics that were present in all people who had Down syndrome. A lot of things are present only in some. But every person with Down syndrome has midfacial hypoplasia or retraction and a relatively brachycephalic skull, wide and short from front to back. And Roger felt if we could show that this mouse model has those physical characteristics, then we're going to be on our way to be able to use this mouse model in biomedical research. And I thought, I can help you with that. So we bred mice. We decided on the bones that, well, we didn't decide. We knew this. Homologous between mouse and human. We found landmarks that could be comparable between mouse and human. And I did a EDMA analysis comparing the TS65DN mouse to their unaffected littermates. And lo and behold, they had maxillary hypoplasia and they had a brachycephalic skull. And Roger was really happy. Now, at the same time, my close collaborator, Mimi Jabs, was creating mouse models for craniosynostosis. And what the diagnostic criteria for this model to be accepted would be to have premature closure of the coronal suture, because that's what Apert syndrome has. And indeed, that was shown easily in these mice. This is P0, that means day of birth. So at the day of birth, they already had premature closure of this suture. But I had always wondered, how could the premature closure of a suture on the neurocranium be causing all this facial dysmorphology that's seen in these kids? This doesn't make sense to me. But we couldn't study humans prenatally. But now that we had a mouse model, we could study these mice prenatally. So what we did was to harvest mice prior to birth. This is at E17.5. That means embryonic day 17.5. Mice are usually born about day 20, 21, depending on the strain. We took these litters and we used microcomputed tomography imaging to do a three-dimensional reconstruction of them. And then again, I just analyzed to find out, are they different from their unaffected litter mates while the suture is still open? And what this is showing you are the lines in pink are linear distances among landmarks that are statistically significantly shorter in mice carrying the mutation, while the lines that are in blue are statistically longer in the mice carrying the mutation. So in fact, yes, they were different. They were much different than their unaffected litter mates long before the suture is closed. Now, please don't understand me. Suture closure is an incredibly important part of this disease, causes a lot of problems, but from this result, it told me that it couldn't really be the trigger for much of the dysmorphogenesis that we see in these children. So we decided we should look at all the tissues that we could. And we used magnetic resonance microscopy and specific contrast agents to visualize the brain, the brain and several what we call negative spaces. So like the the nasal pharynx. We looked at the cochlea, semicircular canals, brain, vitreous humor, and what we found, I won't bore you with the gory details, but what we found were many of these were different also in mice carrying this particular mutation. So we were beginning to amass evidence that these mutations that were on a gene called fibroblast growth factor receptor, or FGFRs, they were affecting many tissues. They were affecting many tissues very early. So then the question became, if soft tissues and negative spaces of the developing head are dysmorphic early in development of these mutant mice, how does this affect skull formation that occurs relatively late? Our hypothesis actually became one of morphological integration, and we were asking questions, how do these spaces, these soft tissues and developing bone impact one another during growth? Now, it was known that a specific variant of fibroblast growth factor receptor 2, FGFR2, were associated with these diseases. We even knew that these mutations were responsible for changing intracellular signaling. But this didn't tell us how these mutations produced cranial dysmorphogenesis. And this is because genes don't make structure. Genes provide instructions to cells to make structure. And other information like mechanical strain produced by growing tissues pushing against one another can be just important and provide information to cells. And so we need to recognize and measure and include the chemical and the mechanical properties of developing tissues when contemplating craniofacial development and dysmorphogenesis. So this hypothesis, or my observations, operates under this hypothesis that morphology is mechanism, and especially change in morphology due to growth. So there are many cranial tissues that we could look at that we're interested in, but we became incredibly interested in the cartilaginous skull that forms prior to any mineralization of any bone. So those of you who study vertebrate paleontology know that the modern vertebrate skull is a composite structure representing the amalgamation of 3 skeletal systems that evolved separately. So this is a cartoon of the pharyngeal skeleton. It's the most ancient, and it developed to support pharyngeal slits and protochordates. The next to evolve is the chondrocranium, and it evolved to support the brain. And these two together form the cranial endoskeleton, which is composed mostly of cartilage. The third part to form, which is a totally separate skeletal system, is the dermatocranium, and it consists of dermal bone, dentin, enamel, and enameloid. And in modern vertebrates, these cranial skeletons more or less fuse together to form what we recognize as a skull. Just like in evolution, the pharyngeal skeleton and the chondrocranium forms before there's any bone. So this is a mouse endoskeleton at embryonic day 12.5. It's been cleared, chemically cleared, so all the soft tissue's there. You can just see through it. And it's been stained with Alcyon blue so that the proteoglycans and cartilage are showing off in blue here. And you can see a really tiny Meckel's cartilage and then the first little cartilages of the chondrocranium there. By 1 day later, we've got a full-blown endoskeleton. You can see this beautiful postcranial endoskeleton, but you also see the nasal capsule, lateral walls, big Meckel's cartilage, the outer capsule, and the back of the skull. And then about a day later, we start the formation of dermal bone. And that's here shown in red. These are frontal and parietal growing over the chondrocranium and mandible forming over Meckel's cartilage. So my colleague Kazuhiko Kawasaki put all of this information together, and he drew this cartoon of the mouse chondrocranium. And it's color-coded in 2 ways. Those cartilages in dark blue are cartilages that are going to mineralize endochondrally, or they're going to remain cartilage throughout the mouse's life. And those in light blue are transient, appearing for only a brief period in time, and they're going to be replaced by dermal bone. And what Kaz discovered was that each of these dermal bones that are going to make up the facial skeleton and the anterior portion of the cranial vault, they appear and develop in tight temporal spatial correspondence with the disintegration of these cartilages, so that the frontal bone ossification center mineralization occurs in tight temporal spatial correspondence with the ala orbitalis, and this is a parietal bone ossification center forming in tight temporal spatial correspondence with the tectum transversum. This is a completely dynamic process, occurs in 3D, really hard to study histologically or with cleared and stained specimens, and we decided we needed to be able to study this in 3D. It took all of these people, a COVID year and then some, to develop the methods for staining, imaging, and segmenting cartilage from embryos. This is what Kate Lasciotto came up with. This is her dissertation work. This is a microcomputed tomography image of a mouse that's been stained with phosphotungsten acid. I hope you can see the beautiful exterior surface of this mouse in 3D, but if we cut sagitally through it, this is micro-CT. That's all of the tissues within the mouse, and here we've just done some 3D reconstructions of some of the bigger organs and superimposed them on this sagittal section. But we were interested, as I said, in the chondrocranium, and we were able to use these data sets. Each mouse was scanned at 6 micron resolution. This is about 2,000 16-bit slice images and about 6 or 7 gigabytes of data for each of these mice. But we now have the morphology, and now we can measure it. So this is what the mouse looks like in situ. We're now, the mouse chondrocranium and the Meckel's cartilage down here, we're now able to segment these out. for all of these prenatal ages and start to study it quantitatively. So here, again, the nasal capsule is to the left, the back of the chondrocranium is to the right. This is a computed tomography, it's segmented from computed tomography. This is a thickness map so that the thinnest cartilages are in purple would pass through yellow, orange into deep red. We also can do the same thing for our mice carrying these mutations that we know have dysmorphic skulls. And I know you're not used to looking at these morphologies, but let me point out just a couple differences. Those cartilages I was talking about before, like the ala orbitalis, is very different between these two. The tectum transversum, very different. And there's this giant deposit of cartilage here in the mouse carrying the mutation that doesn't exist in the typically developing littermate. If you take the landmarks that you can from these samples, and we now have pretty good sample sizes for these. This is simply an Edma analysis. Every linear distance on here in blue is different in magnitude. It's larger to a certain degree. Some of these are 25% larger in this mouse carrying this mutation. There was only one linear distance that was smaller, and the reason that this is smaller is because of this giant cartilage that is deposited here in the mouse carrying the mutation, which shortens this distance between these two cartilages. Now, these observations, as simple as they are important for two reasons. One, what this demonstrates is that a mutation that we have all thought is very active, targets bone cells in terms of morphology. What this shows is that this mutation also appears to be targeting cells in the chondrocyte lineage. It's targeting cartilage. And then secondly, dermal bone, bone of the anterior cranial vault and of the facial skeleton, forms on the outside of these cartilages. So these are not just larger, they're very different in shape too. And because these bones form on the outside of this cartilage, they're bigger too. And they're dysmorphic. They're different in shape from the forming bones in unaffected littermates. So, as I said, if the cartilage is bigger and different in shape, so too will be the bones that are following a plan that's established by the cartilage. And again, in this way, in early bone development, morphology is mechanism. So in summary, what I've shown you is that over my career, I've moved from 2D to 3D in the study of craniofacial growth and development. I've moved my focus from postnatal to prenatal development and trying to understand the production of dysmorphogenesis. And I've moved our focus from bone to cartilage in searching for mechanism underlying a craniofacial disease. But with each of those steps, it was able to use these methods, these morphometric techniques that let me go to the next step and ask the next question. It's been incredibly important in my career to be able to understand these methods and apply them. So I just went on vacation with my husband, and we were in Maine, and there were these yard signs, and I said, "Those are beautiful, you have to take my picture. That's really my philosophy." So if I can leave the students in this audience with a few other my words of wisdom. One, think hard about the questions you're interested in. Find a problem that you're passionate about because you're going to be studying it for a really long time. Let the scientific question dictate the methods you apply. Simple can be good. You don't have to use the fanciest, shiniest method, even though it's fun to do that sometimes. Be kind in your reviews. A review is to help that author make their article better or help that applicant get a better score on their next grant. And what I like to do is to read what I wrote, because sometimes I'll write something and I'm pretty angry at the author. So the next day, read what you wrote, read it back, and think about how you'd feel if you were reading that as a review. And finally, collaborate. I've been able to share stories of some of the people that influenced me the most, but I've been influenced by so many people over my career. I'm sure I'm missing some names here, and I apologize to those that I have forgotten. But don't just collaborate and ask them to help you. Get to know them, get to know their discipline, and let them get to know your discipline. And with that, I want to thank my lab for all making wonderful slides for me. Thank my funding that has been, I've been so fortunate with that, and all of my students, colleagues, and mentors, and especially Dr. Rolfe for having this wonderful prize I'm so proud of winning. Thank you very much.
So I want to thank everyone again for joining us today here for the Rolfe Medal Lecture and in celebration of Jim's philanthropic legacy for the university. And I especially want to thank, we should give our speaker another round of applause for a truly amazing lecture that even as a chemist I found inspiring in thinking about different ways. And I'm really envious of the beautiful images that you've been able to quantitate. It's not just art, but it's really very, very quantitative. Got the replicates there with your animal model, and that's just wonderful. So let's give Dr. Richtsmeier another round of applause. So I do want to express my gratitude yet again to Dr. Jim Rolfe, Distinguished Professor Emeritus in Anthropology and Ecology and Evolution. He's been so generous with his philanthropic investment made to the field of morphometrics, as well as Stony Brook University. He's been a pioneer in this field. He's been a pioneer at the university in the first generation of faculty who really got this university off the ground. And he's made significant investments over the years. In 2013, Dr. Rolfe established this Rolfe Medal Award Fund, which made today's event possible. Then he made a generous commitment to establish the F. James Rolfe Endowed Chair in Morphometrics at Stony Brook University as a legacy gift, and this investment will ensure the advancement of the field and preserve his legacy in the profession. And most recently, this month, I'm delighted to inform you that Dr. Rolfe and his wife, Janice, have established the Rolfe Graduate Endowed Fellowship in the Anthropology Department. And this endowment will support our doctoral students who are working on their dissertations. It's for a dissertation completion award, which is one of the most needed forms of support for our graduate programs and for research. And I can imagine it would have helped our speaker when she got those very unpleasant reviews to have that kind of funding. So thank you to Jim and Janice for making that possible. It's really a testimony to the passion and commitment to morphometrics and to anthropology more generally. I want to just take a moment to speak about the importance of endowed funding, such as the multiple endowments now established by Jim and Janice Rolfe. As a member of the Association of American Universities, which is an elite group of 65 research-intensive North American institutions, Stony Brook University finds itself in a highly competitive academic landscape, and to succeed in this environment in our research requires us to graduate exceptional students and to generate new knowledge that makes a difference in the world. And to do this, we need to have the best faculty and attract and retain eminent scholars and researchers, as they are a lasting tribute to the generous donors. And so the Rolfes have made the lasting faculty, the graduate students, and the research to really stimulate our thinking from the outside. So with that, I just want to welcome Justin Fincher, our Vice President for Advancement, who's going to be encouraging the rest of us to follow the legacy of Jim Rolfe. Thank you, Justin, for joining us in today's lecture, and look forward to seeing you at the ceremony. And lastly, let's just give Jim and Janice one last round of applause for your generosity, leadership, and commitment. And with that, I think we are adjourned to the refreshments in the lobby, and we will see you out there. Thank you.